And thanks to the panel for really dealing so elegantly with the issues we need to overcome and the way forward. The final session this afternoon is a panel on disruptive therapeutic platforms, new tools and new outcomes. We've heard a lot about some of the approaches to identifying mechanistic targets, and I think some of these new targets are going to come up in the course of this last session today, which is going to be chaired by Tony Coles, who's the CEO of Humanity Therapeutics. Tony, welcome to you and the panel, and look forward to your discussion.
Like that.
Well, Callum, thank you for that terrific introduction. Good evening, everyone. We are quite mindful of our place on the agenda and the impending break and networking and drinks. So we promise this is going to be the best panel of the entire day, and there will be lots of learnings to take away. We're delighted to be here with you today. A lot of what you will hear this afternoon speaks to the value and the importance of therapeutic breakthroughs across all of the therapeutic areas, but obviously we're today focused largely on cardiovascular. We've got experts from each of the hottest, most advanced technology areas that are under development today, and so they'll have a lot to share. Leadership in the world, from a U.S. point of view, economically, has largely been built on innovation. We know this. We watch what's happened in the tech space, and we've long been advocates of what's been happening in the life sciences and the biotechnology space as important contributors to the GDP for this country. What we know is that the proof of this economic leadership is really all around us. We're watching mortality rates improve, survival rates improve, morbidity rates decline. All of these things largely in part because of breakthroughs in terms of diagnostics, therapeutics, as well as devices and other technologies. And we sit at the nexus or the crux of the advancement of several important technology-based devices. We're going to be largely focused on therapeutics in this conversation because each person here represents a therapeutics company and a new or disruptive technology. But I start with that broad brushstroke understanding so that we're all aligned on the value of disruption and the importance that it plays, not just in the lives of patients and those we take care of and those who will benefit from our work, but also because of the economic importance of these breakthroughs. We know that cardiovascular disease causes 1 in 3 deaths in the world, and some of what you'll hear this afternoon will spark the imagination, perhaps spark a thought, and a new area of inquiry or research for someone here with us today. Let us now hear from our panelists. I'm going to ask each panelist to introduce themselves and to, if you will, tell us a little bit about your company and a little bit about your area of overlapping cardiovascular interest, if so.
Great.
Stéphane?
Thank you, Tony. Good afternoon. My name is Stéphane Bancel. I'm the CEO of Moderna. So Moderna is a company that is based in Cambridge, which is working on using a messenger RNA as a drug. We are currently a clinical stage company. We have 5 products in the clinic. We just published last week our first human data, which is always an interesting test for new technology. And we are present in infectious disease. We are present in immunology and also in cardiology. We have 1 drug in the clinic in Germany with AstraZeneca, an mRNA coding for VEGF to help patients after an MI.
Good. Thank you, Stéphane. Katrine?
I'm Katrine Bosley. I'm the CEO of Editas Medicine, also Cambridge-based. And Editas is focused on genome editing, and in particular, our platform is based on something called Crispr, which I find that many people have read about in one form or another because it's a technology that's very much captured the public's imagination in addition to scientists' imagination, and it's changed how many research scientists think about biology and what kinds of biological questions they can ask. At Editas, we're here to try to turn that very exciting young science into medicines and tackling all those translational questions and needs to go from something that's cool in a laboratory to something you could potentially give to a patient. So I'm sure we'll talk about what some of those challenges are, but the core of the technology is to be able to very precisely create changes in DNA. And Crispr is this bacterial system that has now been co-opted and repurposed into being able to manipulate DNA in pretty much any biological system. So we hope to apply that in a number of different therapeutic areas. In all candor, cardiovascular is not on the first of the list because there's certain challenges there that aren't the easiest to overcome. But I think the progress we make in our initial disease areas, diseases of the eye, of the blood, of the liver, we'll figure out how to translate this science so that then it can be applied more broadly into a broader set of therapeutic areas.
Thank you. Mark?
Thank you, Tony. My name is Mark Cobbold. I'm an associate professor at Mass General. Formerly I was a clinical immunologist in the UK, and I moved here, and I'm a tumor immunologist and run a translational lab. And my lab really focuses on tumor antigens, the nature of what cancer looks like to the immune response, and mechanisms by which we can alter the appearance of a tumor cell such that it actually matches the immune response that the patient has. And clearly, these both relate to cancer immunology, and there's been a sort of revolution in cancer in realizing that the immune response actually does play a role in protecting us against cancer after all. And so, timing is everything. And for me, landed here really because immunology and cancer are converging in this very exciting field.
Good. John?
Yeah, John Maraganore, Chief Executive Officer of Alnylam Pharmaceuticals. We're a company focused on advancing RNAi therapeutics as a whole new class of medicine. As I heard Katrine talk about the great work at Editas, it reminds me of where Alnylam was 10 years ago, actually, in many ways. A cutting edge, new technology, new science, Nobel Prize winning discovery that needed a lot of work, a lot of effort to figure out the technical hurdles, to figure out how to make these small interfering RNAs, the molecules that mediate RNAi, into drugs. And that's what we've been focused on as a biotech for the last 14 years. And we're pleased that we now have 8 programs in the clinic, and we'll have our first phase 3 data readout this year. So a very exciting point for the company. Now, in the cardiometabolic space, we do have a very interesting program targeting PCSK9, a drug called inclisiran that's about to enter phase 3. We're collaborating with a company called the Medicines Company on that program, and I'm sure we'll talk more about that program during the course of the panel.
We will indeed. Ryuichi?
Hi, my name is Ryuichi Morishita from Osaka. Actually, I founded the company name is Angios at the 1999, the gene therapy companies. Angios has become the first IPO biotech in Japan, and now the company have 8 different levels of pipeline in clinical. The first one is already finished phase 3 double-blind placebo control study to generate angiogenesis in PAD. And hopefully the company will submit the IND in this year to Japan FDA. The name is PMDA. And also in global, the company have now restart the phase 3 trial to treat the CLI, critical limb ischemia patient, including the US and EU and other countries. And recently, the company have also tried DNA vaccine to treat the hypertension. The phase 1 will be started probably June or July in this year in Australia. The company also have tried such kind of gene therapy area mainly. Beside the company, I also have a lab, Osaka University, and more focus on peptide and DNA vaccine to treat adult common disease. Recently, I also work in the government side as a strategic advisor to Prime Minister Shinzo Abe, and also the work in the member of regulatory affairs, including the the law for drugs. That is my point.
Thank you very much. So we're going to start the questioning logically with the immunologist in the group. And Mark, You know, a lot has been said about the future of cardiovascular research. We were able to achieve such incredible outcomes results with biology-based research insights, really understanding what's happening at the tissue level and working to reverse that. We now sit on a threshold where the genome and phenotypic expression will give us even deeper insights in addition to the biology-based learnings that we've got to date. Talk a little bit about why cancer research and cancer therapeutics have been so successful and what lessons we can transfer to cardiovascular therapeutic development. I mean, it's really been astounding. I spent a lot of time in the cancer area, watched breast cancer mortality rates decline. Talk a little bit about those essential lessons and what lessons we can learn in cardiovascular research.
I think there has been a revolution really in the cancer space in terms of how we approach patients. I think one of the things that we were doing wrong initially was really giving patients a single course of therapeutic, and I think the emergence of personalized therapeutics in particular, whereby you would tailor a therapy. Knowing the molecular lesions within your patient's tumor target has brought probably some of the largest gains in health. And so we no longer see as, you've introduced breast cancer, bowel cancer, it's no longer seen as breast cancer, bowel cancer. It's, seen according to the various oncogenes and rearrangements, and even mutations within those oncogenes are targeted. And that way you can actually combine a very personalized approach to a patient's tumor. And most recently, of course, in the advent of immunology, a field that was pretty much completely dead, which was cancer vaccination, has been brought back again. And the knowing that actually, if you can predict the precise mutations within the cancer, and many of those mutations, the vast majority of those mutations, have nothing to do with the cancer at all. They're just passenger mutations, but they're not silent to the immune response. And so with the knowledge of those very specific mutations, you can then create vaccines, and they are effective. So you can then vaccinate the patient with an on-target immunological approach. So I think that's one of the things, the acceptance of personalized therapeutics and perhaps a shift in the way pharma approaches things, not developing one drug for one disease, but actually providing a portfolio of therapeutics which you can then combine in a personalized mix to then target those patients. And I think...
So Mark, I want you to finish, but I want to ask you one question, because you said something a moment ago that triggered a thought. You've essentially talked about a renaissance in immunology research and immunology-based therapeutics. What was the deep insight that caused that renaissance? And can we find a similar renaissance in cardiovascular research?
Yeah, I think the renaissance moment really came because immunology has been bubbling away for 50 years plus, particularly in diseases such as melanoma. But the renaissance moment for I think most oncologists, and it wasn't the immunologists that woke up, it was the oncologists, when they saw therapeutics, immune therapeutics delivering to very common cancers such as lung cancer. Lung cancer was never ever thought to have an immune response or an immune component to it whatsoever. And there was this basket trial where patients were recruited with all sorts of tumors. And as a tumor immunologist, I would have predicted that none of them would have responded. And actually, a very large percentage of those patients responded to those therapies. So what that told you is, in fact, guess what? There is an immune response in patients with cancer, and actually there have been more recent studies that show that the vast majority of patients with cancer have immunity against their own tumors. And so with that realization, you can actually reinvent and try and develop therapeutics that actually says, okay, how do we empower that residual immunity that is against the tumor? And so, yeah, and for me, how this might, and I think cancer is often sort of trickled other fields. So rheumatology, autoimmunity is often sort of benefited from cancer therapeutics. And I think, some of the mechanisms we are uncovering are tolerance mechanisms. So in the cardiovascular space, cardiac transplantation, there's a role where patients need to take very powerful rejection medicines, we're now understanding how tumors prevent the immune system from attacking it. And to my knowledge, I don't know of anybody that's really exploiting those molecules, molecular pathways.
So let's come now to, Stéphane, you've got a VEGF messenger RNA-based therapy that's in the clinic. What's the VEGF as a concept inhibition, at least, has been around for a decade. Talk a little bit about the deep insight there, just bridging the comment about renaissance, if you will. Tell us about the transition and what can we expect from VEGF 2.0?
Yes, that's a great question, Tony. So the story of this molecule is quite interesting. So Ken Chen was a cardiologist, who studied VEGF for a long time. I tried to use VEGF recombinant to regrow vascularization. And the problem with using a recombinant is when you inject it, you need to have such an effect locally that because it goes in circulation right away, basically you end up having no therapeutic index because you need to give a very high dose to get the local effect, but then you have VEGF all over the body. And so this program was stopped. He then tried again with gene therapy. And the problem with gene therapy, as Ken will describe it, is that you actually end up killing the animals faster than the controls because they keep making VEGF all the time, which is not a good thing either. And when you saw the PK of an mRNA, when you inject an mRNA into an animal, is you basically have a huge paracrine-like factor that then 48 hours after, when the mRNA is degraded, goes away. So basically you can get a big spike of VEGF at the site where you inject it, and then 48 hours after it's gone. And because the way we are using this program is in an unformulated, so basically it's in water, what happens from a biology standpoint is that, and this was a great insight from Ken, which is if you inject it into the heart, the mRNA that gets uptake into the heart muscle at the site of injection by the surgeon is going to make for 48 hours around 1,000 copies of VEGF molecule. So we have a very high concentration at the tissue level, but whatever gets into the serum gets degraded by nucleases in seconds. So you have no systemic exposure. And so that was the insight that he had, basically, was combining the PK of this new technology that he saw at the time, in Harvard, in a lab meeting, combining with the expertise he had over 20 years of one pathway, and putting those two things together, saying that is the thing I've been looking for 20 years, is a big spike of protein concentration that then goes away.
So this is fascinating to me. This is a new area for all of us. But what you've just said is we're taking an old mechanism and we're thoughtful both about the delivery and the vehicle by which we administer it. So talk a little bit about those two things in particular. How does the VEGF therapy, the mRNA therapy work? How do you get it into the heart? Just walk us through that, because this is going to be a question that we're going to come to with CRISPR in just a moment.
Yes, so the way Ken tried initially was just direct intracardiac injection into mice, and then when he went into larger animals like pigs, and the data were presented by AstraZeneca last November at the American Heart Association, where basically they show by either direct injection into the heart or using just catheter. Because it's a one-time procedures. So the idea that of the target product profile is, the idea is within a few days or week after an MI, you will go and you will inject once the mRNA, where basically VEGF is going to start revascularization of the heart.
So the infarcted tissue is dead, that's gone,
Correct.
That's not coming back. But the ischemic tissue, the residual ischemic tissue,
Correct.
we think will...
Yes, and so far it's only been proven in mice and in pigs. The early data was presented last fall. There's, I think, a paper being reviewed as we speak. And now the molecule is in phase 1. And the early data is quite exciting. We just saw some early sneak peek at the data 10 days ago. And the AstraZeneca team is working toward preparing a phase 2 early next year.
Okay, good. Well, that's quite exciting. So, Katrine, how do we think about CRISPR as a gene editing technique, the in vivo application versus the ex vivo injection into solid organs, the same questions around delivery and distribution? Talk a little bit about some of the challenges we might face in cardiovascular medicine.
So if you think about what is it you're trying to achieve with delivery, in the case of CRISPR, you need to get this somewhat complicated molecule. It's part protein, part RNA, and both pieces need to show up in the same cell in the same moment in order, and then be able to get into the nucleus of the cell in order to be able to edit that DNA. So what cells do you have to get to? Depends on what disease you're trying to treat. If you're working on, for example, sickle cell disease, then the hematopoietic stem cells are the target cell. You can take those out of the body, edit them outside the body, and give them back. So that's the ex vivo approach that you refer to. But if you can't really do that with the heart. It's a little hard, you know. So as you think about what other tissues you want to get into depending on the disease, you think about other delivery vectors. Now, by and large, there's kind of two big categories. There are viral vectors. A lot of the gene therapy world has developed these viruses where viruses are excellent at getting DNA into a cell. So you can take advantage of that approach, and inside of your viral vector, you put the DNA to make your Cas9, which is the protein part of your CRISPR molecule, the DNA to make your guide RNA, and they're basically saying, shove this into the cell, please use this to make the protein, make the guide, put it together, go to the nucleus, and edit the DNA. That works. It sounds complicated, it is, but it actually does work. And part of the translational science that we work on in companies like Editas and Crispr is to figure out how can you do that and how can you control exposure and get specificity and all these things. So viral vectors are one approach. Lipid nanoparticles are another approach. It's been used a lot in various sorts of oligonucleotide delivery. So we can take advantage of gene therapy over here. We can learn lessons from the oligonucleotide, antisense, and RNA-based therapies to use those sorts of nanoparticles to deliver. You can either deliver DNA or proteins, different things you can use as your construct, your product construct. And then the goal is, can you make it specific to the cells of interest that you're trying to affect? So the delivery is certainly a very significant challenge in this field, and some tissues are easier than others. The easier tissues right now are eye, liver, and blood, but heart is certainly on people's mind. And so certainly the opportunities are also driven by what do we understand about the genome, and whether you want to approach a genetically defined disease where you have a mutation and you're trying to correct the mutation, or what we'll call a genetically treatable disease. And I know we'll talk about PCSK9, it's everybody's favorite poster child. If you're going to make let's go way speculative for a moment. If you wanted to create a PCSK9 genome editing medicine, you're not editing a mutated gene, you're editing a normal gene based on knowledge of the human genome. Now, to even contemplate going there way far in the speculative future, we have to have a very solid, confident understanding of how to make genome editing molecules that are very specific and safe and deliver, all of those technical challenges. And so the kinds of things we work on today in genetically defined diseases, very serious diseases with no other good therapies, et cetera, does lay a foundation for someday reaching out to what I'll call genetically treatable diseases, and certainly in therapeutic areas where delivery might be more challenging, such as the heart.
Well, we're going to come to PCSK9 in just a moment, because that has certainly taken the cardiovascular world by storm. Katrine's reference to CRISPR just a moment ago was not the technology, it's Crispr Therapeutics, one of the other companies. And we've got a wonderful friendship. She's the CEO of Editas. I'm the chairman of the board of Crispr Therapeutics. So like the previous panels, so many of us are touching this really exciting technology. Ryuichi, in just a moment, we're going to come to you and we're going to ask you a little bit about oligonucleotide therapy and specifically gene therapy. And along the same line, because so many of these technologies sound great in theory, look good on paper, but we really have to be practical and thoughtful about how we get them into the body. But before we get to you, John, we're going to come in to you and have you talk a little bit about PCSK9. Talk a little bit about the collaboration with Medicines Company, talk about the program, talk about whether you think you can actually make a difference, a big difference in this area.
Thanks, Tony. I mean, the PCSK9 as a target, I don't think needs much of an introduction. I mean, it's a genetically defined target that's involved in regulating the LDL receptor. There are gain-of-function human mutations that are associated with hypercholesterolemia. There are loss of function and even null mutations with PCSK9 that are associated with decreased levels of LDL cholesterol and beneficial effects on coronary artery disease. So the target itself is really one of those sui generis disease targets where the confidence around it as a target for an antibody therapy, which is, of course, the currently improved therapies, or for that matter, more advanced therapies like RNAi, the target itself needs no discussion, no introduction based on the human genetics. We entered the field of targeting PCSK9 pretty early in 2008, and our first foray, and this, by the way, is an interesting example of how many advanced technologies ultimately have to mature. Our first foray was in lipid nanoparticle targeting PCSK9, and we showed in a phase 1 study that we can get robust knockdown of PCSK9 and that we can lower LDL cholesterol, and we had an IV-based approach for targeting PCSK9. Well, guess what? Hardly competitive, hardly differentiated. At that time, there were 4 other antibody programs that were ahead of us in the clinic, including Amgen, Pfizer, Lilly, Novartis, actually 5, Regeneron, and Sanofi. So pretty crowded field. We then developed internally advanced technologies that enable sub-Q delivery with incredibly durable knockdown effects of target genes with our GalNAc technology. And we deployed, starting back in 2012, we deployed that technology on our PCSK9 program and developed a drug called inclisiran, which has now been in 2 separate clinical studies, phase 1 of around 80 patients, a phase 2 that just completed of around 500 patients. There are 2 New England Journal papers that describe both of those studies, and we've been able to document a really impressive lowering of LDL cholesterol of over 50% with a subcutaneous dose regimen that is either quarterly or potentially biannual. And that then creates a level of differentiation from a product perspective that is really compelling in the overall
So I'm going
space.
to, there's a question from someone in the audience.
Yeah.
It says, given the commercial performance of Amgen and Regeneron's anti-PCSK9, do you think that the market for yet another anti-PCSK9 exists?
Well, I'll start by saying that, 3 of the other players that were in the space left the space, okay?
Because you got in.
No, we've not. But they left the space for different reasons. Pfizer made a concerted effort to get to the market with their antibody, but their antibody ended up having antibody drug responses and therefore creating an antibody whose effect waned over time. And so they discontinued their program. Novartis and Lilly sort of stayed on the sidelines as this whole world advanced. Now, commercially, Sanofi, Regeneron, and Amgen are commercializing their products, and the products have been met with headwinds around reimbursement at the end of the day. I do think, and I think many people in the field believe, that those headwinds will relax as more outcomes data get presented. The Fourier data that were presented at ACC clearly provided the beachhead for those type of outcomes results for the Amgen program. And I think we can all expect at the American Heart Meeting that Regeneron and Sanofi will present data from their outcome study, which will be equally supportive, if not even stronger, because they had an enriched patient population for higher risk than the Amgen study. So I think this is a class that will take time commercially to advance. But what's great for us is we have a drug that's a biannual or quarterly sub-Q drug, which really can be disruptive in the space, because the number one issue, or a major issue in the space, is compliance. Patients are generally non-compliant with their statin drugs. About 50% of patients stop taking statins within a year of their first prescription.
Well, that's a clear advantage. A once or twice annual injection is a clear advantage. But what should we be thinking about in terms of off-target or tolerability issues with this technology?
Yeah, so in the 500-patient phase 2 study, the adverse event rate in placebo versus drug, but for injection site reactions, which were mild and not really problematic, were indistinguishable between placebo and drug. So far, so good, Tony. Now, the phase 3 study, which just got endorsed by the FDA, will be a 3,000-patient study, so a little bit larger, not much larger, but a little bit larger, with LDL as a primary endpoint. And there will ultimately need to be a CBOT study, which will be a 14,000-patient study.
And you've studied how many patients to date?
To date, about 580.
Okay.
Yeah.
Okay, good, good,
Yeah.
good.
So far, so good.
So far, so good. All right, my money's on John.
Thank you.
Talk to us a little bit about gene therapy. You know, this has been the promise that we talked about a moment ago in terms of, you know, we have a lot of insights from biology-based drug research. The promise of gene therapy has just been outside of our grasp. It's a little bit like cancer vaccine therapy. Talk a little bit about how we overcome the limitations and the barriers for the technology, and then we want to talk about cardiovascular specifically.
I think that in the difficulty of gene therapy is still the medical device, how to affect to the heart. So people, especially we and others, have used medical device to transfer directly into the myocardium by casters. But the caster technology is not well. So sometimes, you know, it is very difficult to transfer directly. So then people have moved to the new medical device to transfer AAV vector into the myocardium via the coronary arteries. But still, we don't know how exactly affect, especially to become the real beneficial outcomes. Currently, only we have a very good technology to transfect the muscles using AAV vectors, such kind of glibera, the transfection of the lipoprotein lipase. That is approved in EU. Unfortunately, that drug was drawn because only one patient was treated. But definitively, you know, a vector is useful to transfect into the myocardium. Therefore, gene therapy area of cardiovascular disease, now it is possible to increase some kind of recombinant protein from the transfected gene in the muscles. I think that's one possibility, probably to treat Fabry disease, mucopolysaccharide, and other kind of disease. And in other areas, I think the DNA vaccine or AAV vector vaccine might be more useful because it is easy to increase antibody, so to antagonize PCSK9 or other kind of vaccines. I think that is a possible areas.
One of the programs Angis has underway is in critical leg ischemia. So talk a little bit about that program, and given what you've just said about intramuscular therapy, talk a little bit about that.
Okay. Actually, you know, on this program, we transfect directly plasmid DNA into the muscle of a critical limb ischemia patient. We used hepatocyte growth factor gene. The angiogenic growth factor activity is very similar to VEGF and FGF. And we conducted the placebo double-blind study in the United States as well as Japan. Especially in Japan study, we got a very good result to decrease resting pain as well as ischemic ulcer. So now Japan government have changed the law for drugs. So we have accelerated and conditional approval systems, especially for the regenerative medicine, including cell therapy and gene therapies. So using these systems, company have tried to submit NDA into the Japan FDA soon. But except of these programs, we also tried into the myocardiums under the PI, Dr. Mike Simons. The preliminary result as a phase 1 seemed to be very well. But we found that, you know, again, custom systems has many kind of troubles in clinical. So therefore, we stopped these programs. Now focus on the CLI. But hopefully, if we found very good, you know, vectors or some medical device, we want to again, you know, enter to this field, especially to treat chronic heart failure or ischemic heart disease using hepatocyte growth factor.
All right. Well, we still have a lot to get done. We're just over 10 minutes left in the panel, so I'd encourage audience questions. We've got one here queued up for Katrine, which will just come. But a little bit about the ground rules for the next part of the panel here. So, Mark, you know, when I trained at MGH, 25 years or so ago, the immunologists were the nicest guys. You'd call them if you had a hot joint or if you needed a consult on that really difficult to treat RA. We're going to put you in the prime position today, meaning you're going to judge how well we're doing as a group in terms of the renaissance in cardiovascular therapy. So you're going to get one of the last words. You got the first word, you're going to get one of the last words. You got a huge promotion there. So it's going to be on you. We're also going to ask each panelist to tell us something. I love the Sunday talk shows. I don't know about you guys, but I love the tell us something we don't know. So you guys are going to get to tell us something we don't know. But Katrine, here's your question. Viral-based CRISPR delivery in the cell. Does viral particle-packed CRISPR have any effect on immunological response in humans?
Excellent question, and we will find out. It is a great question, and I think clearly we're working with a bacterial protein here, and I think there's a few considerations. One is that the protein is going to be made inside the cell, so you do have to consider, is it possible it could get presented, and could there be an immune response from that? So, certainly if the body were exposed, systemically, we would expect that the immune system would respond to a bacterial protein. But, in intracellular delivery, will there be a response or not? There have been, in the literature, lots of different folks who have done various sorts of animal models at this point, and that's not that's anecdotal in that it's not a thorough sort of GLP type assessment, but there's certainly, we don't see a sort of florid immune response. I think too that one of the things you have to think about is what exactly, as we think about where could you go with viral vector delivery over time. And in some cells, it might be fine to just have the delivery, and with AAV, it might persist for some time.
AAV being? I'm sorry,
adeno-associated viral vector delivery, which is you mentioned it earlier, it's being used quite a bit in gene therapy, and it's a good tool for this sort of viral vector delivery. But we're also going to want, as we develop more advanced forms, we're going to want control over exposure, which I think also speaks to this question. And one of the more advanced engineered forms of the CRISPR molecule that we use is one that we call it self-inactivating, or it's essentially you're able to get control over the time course of expression. So you can take advantage of the natural CRISPR activity and basically allow it to express itself for a while, and then essentially give it the tools to turn itself off. So as you look at the more, where you go long term with real therapeutics, and you start to solve these fine detailed questions of making medicines, not just doing cool science in the lab, these are questions: immunogenicity, PK/PD, what is the control of exposure, and creating variants and variations that really bring those under control. So those are some of the sorts of things that we're working on and have made good progress on, as well as you take into account as you think about what diseases to work on and how do you either minimize or hopefully eliminate some of those risks.
Okay, good, thank you. I'll only point out that Mark is now on the edge of his chair, and he's taking very careful mental notes. So we're coming back to Mark. Stéphane, another question from the audience. Is mRNA safe enough to be dosed chronically? What concerns have kept chronic mRNA therapies out of clinical trials?
Yeah, it's a great question. So the way we think about safety of our technology is around the safety of the mRNA, the safety of a formulation, and the safety of a protein that the body is going to make based on the mRNA we're going to give to the subject. So we've done a lot of work on the mRNA, the safety of the mRNA, and we think that now we're getting to a pretty good place. 500 subjects have been dosed around the world, and we'll keep improving that piece. The safety of a protein is, of course, depends on the protein that you dose, which is why we did not start with diabetic and insulin, because as we all know, too much of insulin is not a very good thing. And so we start by the diseases where there's huge therapeutic index. So if we give too much, it's going to be okay. And then you go to the formulation. I mean, John spoke about formulation, and they have a lot of experience in formulations. What we've had to overcome is the ability to give a formulation that has a short half-life. The formulation that we started with were not biodegradable. So one issue that we saw from a safety standpoint is that if you were to dose regularly and go back, like for, let's say, a rare disease, you would end up having accumulation of that non-biodegradable material coming from the formulation, not the mRNA, or would kind of accumulate into the tissue, which will not be a good thing. So this we solved because we have a new generation of formulation that instead of having kind of 6-day half-life, which is what we had when we started the company. The current technology, which is going to be going in man later this year, has a 2-hour half-life once injected into the body. So we think,
is that sufficient?
So we think for a lot of diseases it is sufficient. For some it will not be sufficient. But for a lot of diseases we're contemplating now, we think it's going to be kind of a bi-weekly injection. So if you inject bi-weekly and 2 hours after, a few hours after you inject, the formulation is gone. It has been shown in different safety studies that we have run that this is fine in a primate or into a rat. So what we want to do now is to gear up to do GLP tox study and to show that in human. We should have a paper coming out pretty soon that I think will look at 7 or 8 doses injected actually twice a week, because we try to do it kind of short intervals, where we did that work with AstraZeneca. So it will be a co-authored by the AZ scientists and our scientists that shows that you can dose repeat the technology, at very short intervals for quite some time without safety.
So in lieu of almost a depot formulation with a long dwell time, you'd rather inject more frequently in the hopes that the material will biodegrade.
So it really depends on what you're trying to do. So you really have to think about it molecule by molecule. Okay. But again, I think the first repeat dose molecule we're going to take to the clinic are going to be, I think, every 2 weeks type of treatment with a very short half-life on the formulation, which is great, we think, from a safety standpoint. And only later on, when this is going to be understood in the clinic, we might look at other therapies where you need to inject because of a half-life of a protein and because of a disease you're trying to cure on a shorter dose. But again, we're trying to learn to work. I mean, you hear the same theme across the panel, which is with new technology. It might be very noble to try to go something, very crazy right away, but this is how you might die and be done with it. And so we're trying to always find the space where we think it's safest with what we know, because we don't know what we don't know of the new technologies we're all experimenting in animal and then in man, to be able to learn to work before we're going to go for a drug.
Well, then we know we've got the right group, because that's where disruption starts. Okay, so panelists, the question is to tell us something we don't know, and we're going to actually start with John. Tell us something we don't know.
Well, Tony, I don't think we yet know how the disaggregation of, staying in the theme of cardiovascular disease, the disaggregation of cardiovascular disease based on targets, biomarkers, genomic information, will actually facilitate the opportunity for these more advanced medicines to make an impact on those diseases. I think we're at the beginnings, really, of understanding how disease can be dissected into larger, smaller groups, larger number of smaller groups, and drug development in those areas will be very distinct from drug development in traditional cardiovascular type studies. And I think they'll be well suited for these type of more advanced medicines, whether it's RNAi or CRISPR-Cas9 or mRNA delivery. So I think there's a big opportunity in the future for advanced medicines platforms to emerge, even in these large disease indications, when those large disease indications become smaller diseases, which they really are.
So let's bridge to Stéphane for a second, because we've got another audience question. Stéphane, will VEGF overexpression lead to a non-functional leaky capillary system developing in the heart?
Yeah, it's a great question. What we have seen so far, and I want to be careful because we're still at the beginning of this program, is that this is very dose dependent. And so like you do for a lot of molecules that we have all developed through the years, is you do a slow dose escalation, you understand the PK of a molecule, the PD, and you're going to have to find the right window. But the scientists have been able to do that both in mice, in rats, and in pigs. And so we are hoping we can do the same thing in man, but this has to be demonstrated. We have already run 2 different cohorts in a single dose escalation phase 1. The third cohort is going to start dosing very soon. So we're going to get a good sense, and then we'll have to see into a phase 2. But I would say this is like any drug development process. Is you have to go step by step and be careful, because as I said earlier, you have to think about the mRNA tox in our case, the formulation tox. You should not forget the protein tox. That's a good example where you might have a very nasty protein. too much of VEGF might not be a good thing.
Yes, well, that to be determined. Ryuichi, tell us something we don't know. Tell us something we do not know.
Okay. So I think that the remaining issue of cardiovascular disease is some of population science. As you know, hypertension, hypercholesterolemia, and diabetes is still big problems in the developing countries. So recently, I think that the DNA vaccine or some vaccine technology may decrease such kind of cardiovascular mortality in different countries. For example, our company have tried to develop a DNA vaccine against angiotensin II. The system is very similar to angiotensin receptor blockers. But after taking this kind of DNA vaccine, the population, blood pressure of population might be decreased 5 millimercuries. So these some small kind of reductions already estimated to decrease the cardiovascular mortality and morbidity over 10%. Actually, these kind of idea might be another, big issues for cardiovascular disease, especially in different countries.
Good, thank you. Katrine, really quickly, tell us something we don't know.
How many different creative ways there will be to edit the genome? And we've mentioned PCSK9 as a beautiful poster child of understanding the genome has led us to thinking about somewhere in the future that as a target. But a more proximal target that's a good example of just knowing the biology of the human genome is really powerful is hereditary persistence of fetal hemoglobin. So nature has done this experiment where people who continue to express fetal hemoglobin and also have a sickle cell mutation do fine. And so most of us, our fetal hemoglobin is suppressed. The gene editing opportunity there is, could you take the brakes off of fetal hemoglobin in a patient with sickle cell? You're taking advantage of the knowledge of human biology that nature has done the experiment and applying it in a way that's therapeutically beneficial to somebody with, in this case, a genetically defined disease. So it's a beautiful story of biology. Will there be a lot more opportunities like that? I think will be fascinating, because that's not about fixing the specific mutation, but it's about using the biology and the knowledge of the biology to create a therapeutic benefit. How many of those opportunities will there be?
Okay. Stéphane, 15 seconds or less, and then Mark's going to grade us.
So at the end of this year, we're going to start doing a clinical trial in oncology for personalized cancer vaccine, where we're going to make a product for one patient. So this capability will be in a clinic later this year, and we are starting to explore it in other therapeutic areas. So the thing that would be interesting to think about is in cardiology, how could this be applied that you could really make a medicine with one mRNA or 5, 6, 10 mRNA in a single vial to hit any pathway you want that is specific to the genetic signature of that one patient.
So true personalized medicine.
One vial for one human being.
Okay, good. Okay, Mark, the Renaissance.
The Renaissance. Well, as an immunologist, I'm look with significant envy, I think, at the cardiology sphere. Because you kind of solved most of the problems. I mean, if you get hypertension, the drugs are there. If you get heart failure, the drugs are there. You can transplant the heart. So much isn't known in terms of how the heart functions, electrophysiology, development, so fast, so on. And also, I think in terms of how the general public also understand their cardiovascular health. Pretty much everybody I know knows about hypertension and hypercholesterolemia. So I think, you've gone so far with your ability. The question is what comes next, and understanding basic fundamental diseases like type 2 diabetes, metabolic syndrome. At least to an immunologist, I have no clue as to what really those diseases represent. So I kind of at one end would want to grade you an A+. As a community, but the other thing, it's not all solved yet. And for instance, this week, I read a lovely paper showing that actually one of the most important cells in the heart in terms of coordinating cardiomyocyte function was a macrophage. So macrophage, an immune cell coming in, actually coordinating how heart muscle cells.
See, it all comes back to immunology.
It all comes back.
So we're going to take the A+, and on that note, we're going to thank our panelists for an outstanding job.